Welcome to Easy Bio. Today we are going to talk about size of specimen. Size of specimen, how can we calculate the magnification, the size of biological specimen. So what do we mean by that? The size of structure or an organism is measured in units of length, such as millimeter or meter. When a diagram is made, it may not be easy to show the correct size. In this case, this correct size is called actual size. For example, if we are trying to prepare a slide of animal cell, and we can see very clearly the animal cell under light microscope, as you can see, and you are trying to draw it to show off the structure of this animal cell. So you will draw it, but will you draw it with the same actual size under microscope? Actually, no. Even under microscope, we are magnifying it. We are magnifying the cell to be clear, because these are very, very tiny structure, microscopic structures, cannot be seen by our eyes. So they are magnified. So we have actual size or length, and this is the true or the correct size of the cell or the organism, whatever. And we have magnification, and this is how we magnified this actual size to be clear, to be seen clearly. So if I see the animal cell and I'm trying to draw it, I'll draw at the same size like this diagram. Okay, so here from this point to this point, I have the length of this animal cell, which is magnified, and this is called observed length. So the structure or the organism is magnified, and we can calculate the actual size of it by using the equation. We have a specific equation for the actual length. How can we calculate the actual length? By measured or observed length over the magnification. Very important to know this, and any question asking about this, you have to mention the equation, especially in paper 6. In recent exam, this equation or this kind of equation is very common in the recent papers, so it's very important to know the equation and to know how can you calculate the actual length or any one of them. To simplify this, we have a common triangle. Here we have the observed length, we have the magnification in this side, and we have the actual length. Okay, so in case of our example, I saw the animal cell under microscope, I draw it on the paper, and now the teacher asked me to calculate the actual length. So I have the observed one, which is drawn by myself, and this can be measured using the scale, or the ruler. Magnification mainly given to us in the question, and then that's what we need to figure this out. So whenever you have usually in this kind of questions, you will have 2 points which are known, and one of them is unknown, and that's what he's asking about. So if he's asking about the actual one, so you will have the observed, and it will be over magnification to get the actual. If you have the magnification, okay, and he's asking sorry, if he is asking about the magnification, so you will take the observed over actual and so on. So using this this triangle is very important. Okay, for magnification, what are the units? The magnification unit is times, and usually it's found before the number. Okay, not as the other units. We are using the whole units after the number itself, but in case of magnification we are using the unit before the number. Okay, actual length, millimeter or micrometer. This is in the new syllabus. And the key trick of these points, the observed length and the actual length should be of the same units. Don't forget this. So if you have, for example, the observed unit, or you measure the observed unit using the ruler, and you figure them out by using centimeter, so your actual length after calculation should be with centimeter. If you measured it and using centimeter, you have to convert into millimeter or micrometer, then the actual length will be at the same unit. Okay, if we are taking one example, this diagram is given okay, and this written here, and he asks to calculate the actual size of the head of the virus. Actual size of the head of the virus. So what are the steps? What are the steps? First, we have to measure the length of the head on the diagram itself, which is observed length. So we'll take from here to here. This one will be measured. So how much after measuring? It's 17 millimeter. Second, convert it or not, it's up to you, or according to the question itself, to micrometer, because in paper 2 sometimes he's asking about with the micrometer unit. So we'll convert it into micrometer. Then third one, what do we have according to the diagram? We have the magnification, which is 2500. Okay, then fourth one, write the equation. Actual length equal observed over magnification. So 1700 over 2500, which is 6.8 micrometer. But sometimes we use the scale line. What do we mean by the scale line? For example, if we have this diagram, okay, and there is a scale line drawn, and on the scale line you can see 1 millimeter. So, and we have 2 points. Between these 2 points we need to know the actual length of this structure. Okay, first point, I have here scale line. It's written 1 millimeter, but actually when we are measuring this length, it's not 1 millimeter. So according to the scale line, we can get the magnification. How? Actually we have 1 millimeter. 1 millimeter, this consider as the actual one, and this the observed one. So the magnification will be observed over magnification, sorry, observed over actual. So it will be, according to my measurements here, observed equal 2 centimeter, which means 20 millimeter. So it will be 20 over 1. So the magnification is 20 times. It means this structure is magnified 20 times to be seen like this. Now I have magnification according to our work, and I have this one, which is observed for this diagram. So according to our measurements, the observed equal 4 centimeter. Sorry, 4 centimeter, which is 40 millimeter. So now I need to calculate the actual length of that diagram. It will be observed over magnification, means 40 over 20, 2 millimeter. It means this actual length of this diagram is 2 millimeter. This is the whole points in the size of specimen. Thanks for watching.